here do the mitochondria, and then actually go into the mechanism. So there's two parts for photosynthesis. We have what used to be called light reactions, so we could say light-dependent reactions, and then light-independent reactions used to be called dark reactions, but also are called carbon reactions or the Calvin cycle, or we'll call them light-independent. So why should we care about photosynthesis? A lot of times with 211, I usually have a lot of students who want to go into medical or health fields, and they're kind of like, why do we need to learn about plants? Why should we learn about photosynthesis? But this is really connected to human health in many, many ways. So first of all, our entire food supply is dependent on photosynthesis, right? At the base of our food chain is plants, the producers, right? So they are dependent on photosynthesis. Our energy supply, so if we look at coal, oil, that is all made from ancient plants, so we depend on it for that reason. And then just the fact of global warming, that plants play such a critical role in decreasing CO2 levels in our environment. So obviously, we should care about this on multiple different levels. So going back to our equations, I think we never in class talked about the equation for cell respiration. But does anybody remember the overall equation for cell respiration? And then we'll put up the equation for photosynthesis. Yeah.
Glucose added with oxygen.
Yeah. Okay, so glucose, this is for cell respiration. We have C6H12O6, glucose plus oxygen gives us CO2 and water. And we could add in these, right? So that's cell respiration. How about for photosynthesis? What are the reactants for photosynthesis? Opposite. These two are going to be over here. So we have CO2 and water, right? And then it produces sugars, glucose, C6H12O6, and oxygen. And we have to add in light, right? That provides the energy. So these are very closely related. So the products, what we're breathing out, what we're respiring is being taken in by plants and converted into the sugars. We're consuming the sugars from plants. So we are all tied together very closely. The water, of course, the CO2 comes from the environment. The water is being taken up by the roots of the plant. The light is being collected by the leaves, and then they're producing glucose and they're giving off oxygen. So I have this sort of picture I found on Google, and of course they didn't even spell glucose right in that picture, but that's what's happening here. So we just put up the reactions. What is going in for photosynthesis is CO2, water, and photons would be the particles of light. What is coming out of this is glucose and oxygen. And where is that taking place in the plant cells? In the chloroplast. Okay, so that's the specialized organelle for that. Respiration is just the opposite. Oh, we forgot to include ATP on this side. That ATP is produced. And obviously that is happening. If we're looking at bacteria, it all happens in the cytoplasm, but if we're looking at in eukaryotes, it's happening in the mitochondria. So one thing I want to emphasize, since we have talked about oxidation reduction, that both of these are giant oxidation reduction reactions. If we're looking at photosynthesis, we're taking CO2, which is completely oxidized, and we're adding electrons to produce a reduced glucose molecule. The water is going to be oxidized to form oxygen. And then the reverse is happening when we're going through cell respiration. In this case, we're starting with glucose and we're oxidizing it to give off energy all the way down to CO2. And then the oxygen is being reduced by taking the electrons from the electron transfer chain. So that's big picture how all of this fits together. So in this, you can see the central molecule is always glucose. Glucose is either being used or it's being produced. And so why is it that glucose is so central to most of our metabolic pathways? Well, it turns out it's a very versatile molecule. It can be converted into pyruvate, which can then be used to go through cell respiration, create ATP. It can also be a storage molecule. So we can use glucose if we have too much of it. We package it into the form of glycogen or starch if we're talking about plants. It can be used to make nucleic acid. So this pentose phosphate pathway, you're going to learn about that in biochem. It can be shuttled off to make ATP, NADH, these are all nucleic acids, or ribose or deoxyribose. And then also, we talked about structural purposes for it in polysaccharides, like cellulose, chitin, peptidoglycan, right? So it can play a role in structure. So photosynthesis is going to create one of the most versatile molecules that we have here. Okay, so now let's talk about the process of photosynthesis. Where does it take place in the cell? We said already that it takes place in the chloroplast. So I want you to see, rack your brain and see if you can remember what the inside of the chloroplast looks like. Do you remember? Remember there's little discs? So try to think if you can label, what are these membranes? What is each of these little sacs called? What is the whole thing called? I don't know. Take a minute and see if that's still in your brain from, I think we talked about it, very first part of this quarter.
Inner outer membrane? I don't know. Inner and outer membrane? The thing that she pointed. There's 3 types of So look at it. Okay, there's a stroma, there's a grana, and there's a helicoid.
All right, so
This
if we're starting from the outside, working our way in, just a reminder that our chloroplast is one of our organelles that has a double membrane, just like the mitochondrion. So these have the same names as they did for the mitochondrion. We have outer membrane and inner membrane, right? Those two. Anybody remember the name of the sac? What is it called? I heard you say it. Thylakoid, yeah. This is a thylakoid, and we have usually stacks of thylakoids. Anybody remember what that's called? It starts with a G. Yeah. So one stack is a granum, and then when you have multiple, you call them grana. You have plural. Anybody remember what the space in between basically the inner membrane and the thylakoid is called? It starts with an S. Stroma. Yeah. This is the stroma. And then we also name, the membrane itself is called the thylakoid membrane. And the inside of that depends on the textbook or reference. Sometimes they call it the thylakoid space, sometimes the lumen. So either way. Thylakoid, lumen, I forget which our textbook calls it, but okay, so those are all the parts. Now I know for a fact that one of my favorite questions to ask on the final exam is to draw a mitochondrion, label all the membranes and spaces, and draw a chloroplast, label all the membranes and spaces. So you should be able to do that. Where I don't provide it for you, I mean, it's pretty simple to draw the parts for each. Okay? So here's the structure of the chloroplast. Again, this is showing you an electron micrograph so you can see sort of what these stacks actually look like. The liquid matrix is shown there, which is where we'll look at where the carbon reactions are going to take place. So just like we did with the mitochondrion, we labeled where the different parts are going to take place. For photosynthesis, we just have 2 parts. We have the light reactions. They're going to take place in the thylakoid membrane. And then we have the carbon reactions. They're going to take place in the stroma. So those are the only 2 places we have to label. Okay, so to talk about photosynthesis, we really have to talk about the photo part of it, right? So the light part. So just a very quick review on electromagnetic radiation. In the process of photosynthesis, we're converting light energy into chemical energy. So the light energy here gets converted into stored chemical energy in the form of glucose. So we talked about light as being both a wave and a particle. Kind of review, if you've taken physics, you probably know way more in depth about electromagnetic radiation. The basic definitions we need for this class, though, are wavelength and sort of understanding which wavelengths have more energy. So for, what would you say is the definition for a wavelength of light? Yeah, can you say louder? So frequency is related to wavelength, but how would you define, like, if you're looking at a wave like this? What's the
It's the length, it's the space, it's how long it takes to get from one peak to the other peak.
Yeah, or peak to peak or trough to trough is the distance between those peaks, and they are related to frequency. There's all these equations relating energy and frequency, wavelength, and all of this. We're not gonna worry about that for this class, but you can see here the visible light spectrum ranges from about 350 nanometers up to 750 nanometers. That's the visible light spectrum. Just to kind of understand how this fits into the overall electromagnetic spectrum, you can see things like X-rays. Any of you, I mean, we all probably have had X-rays before in our lives. This is getting much smaller, so those are shorter wavelength. Longer wavelength includes things like infrared, microwaves, radio waves, as you get towards this end. And you can have things that are as big as meters, like their wavelength is meters long. Which one has higher energy, shorter or longer? Shorter, right? And that makes sense. So if you go to get an X-ray, it's able to penetrate through your tissue. That's got a lot of energy. So that one should say shorter wavelengths have greater energy than longer wavelengths. And just to place some of the things, other things, just interesting things, on the spectrum, there's our visible light spectrum, and going down things like PET scans, X-rays, and then longer, so we have cell phones, showing you how big the wavelength is for cell phones, ultrasounds, or wireless data, etc. Okay, so when light is absorbed by the plant, which part of the plant is doing the absorbing of those photons? Yeah, the leaves, right? So it's the cells that are in the leaves that are going to be doing most of photosynthesis. And it's specifically, in this class we actually don't go into sort of the structure of a leaf, and maybe I don't know if you guys took 160 in person. Did anybody take it in person and get to do like microscopes with leaves? And that's too bad. But there's cells called mesophyll cells that have most of the chloroplasts that are going to be doing most of the photosynthesis. So those are the cells that are going to be doing absorption. And when the light hits that leaf, there's specific molecules that are able to absorb the light. And those are called pigment molecules. And on the next slide, I'll show you what are the major pigment molecules in leaves. But those pigment molecules will absorb photons of a specific wavelength, and then they either transmit or reflect all of the other wavelengths. So does anybody know, for a green leaf, which wavelengths are being absorbed and which ones are being reflected? We can start with which ones are being reflected. So those are the colors that come back at you and what you see. Everybody remember the spectrum here? So Roy G. Biv, right? The red, orange, green, blue, and purple. So which ones, you can actually, if you don't remember or don't know, you can see which areas are light is absorbed, which of the wavelengths get absorbed by leaves.
Violet, blue, and red.
Yeah, it's the ends of the spectrum. So the reds and the blues get absorbed. In the middle here with greens and yellows, those ones are reflected. This explains why leaves appear green to us because that is the wavelength that is being reflected back to us. The other wavelengths, when they get absorbed, the electrons, we'll talk about what happens during absorption, but the electrons themselves are going to excite electrons in the pigment molecule to a higher energy state. So the pigment, going back to this one, the options for when wavelengths, when light is hitting any object, it either is going to be absorbed, transmitted through, or it can be reflected. Okay, so when something is absorbed, it's actually going to excite electrons in that molecule. If it's being reflected, it just comes back at you. Yeah.
I have a question, but it's not specifically
That's fine.
like the plants, but does sunscreen reflect or absorb? How does sunscreen work with you?
Yeah, that's a good question. I know there's different types of sunscreen that you can apply. So I don't know that, I actually don't know the answer to that. And I know they say to avoid certain types of sunscreens, or anyway, I don't know the answer to your question. I can look into if it is preventing the absorption or how it's actually working.
The physical blocker will like reflect it back and
Yeah.
bounces it, but the chemical one, I don't know what it is.
Yeah, and that's interesting too. Like I know when I went to Hawaii, they had reef safe sunscreens that they don't have the metals, and so I don't know how they work in comparison to the ones with the metal. So we can look into that. That's a great question. Okay, so when they realized that the leaves were doing this absorption, they ground up leaves. This is sort of the historic discovery of pigments, and they used a thin layer chromatography technique where they basically resolved these. And they were able to see the different pigments that are found in leaves. So the most prevalent pigments that we have are chlorophyll. And you've probably heard of chlorophyll. Chlorophyll A and B, there's two different types. Those are the ones that absorb mostly in the blue and the red region. But they also realized that there's some underlying pigments in leaves as well. So things like pheophytin, carotene. So where have people heard of carotene before? And carrots, right? And xanthophyll. So there were these accessory pigments as well, but primarily the major pigments in plants were the chlorophylls. And as you can see in the spectrums, the green lines here, those show where chlorophyll absorbs. So chlorophylls absorb primarily in the blue and the red region. The other accessory pigments like carotenoids, they absorb in a slightly expanded region. You can see that they absorb more into this region. They reflect reds and oranges. So if you think about foods that are rich in carotenoids, what carrots, a lot of red peppers, things like that, they tend to be red in color because they're reflecting in that end of the spectrum.
So plants that are purple,
Yeah.
are they reflecting purple that is?
Yes. Yeah. Exactly. Okay, so if we actually look at the structure for these chlorophylls, we'll see that the chlorophyll molecules, you can see they have this hydrophobic region here. They are embedded into the thylakoid membrane. So they're sticking into the membrane. They have the hydrophobic region that sticks them into the membrane. And it's their electrons that get excited when the light is going to strike the chloroplast. This just shows you a couple of examples of other structures of different pigment molecules. Heme, for comparison, heme is in your blood, and this reflects in the red range. That's why it appears red. So these reflect, whatever color you see with that pigment is the color that's actually being reflected, and the other wavelengths are the ones that are being absorbed. Okay. So if we look at plants, we said they can absorb in the red wavelength, in the red wavelengths and the blue wavelengths, but they're not absorbing in this middle wavelength. And so there's an explanation for that, and it has to do with the amount of energy that's found in that wavelength. The amount of energy that you have, let's say for this, I guess the red one is less energy because it's a longer wavelength. It's enough energy to get the electrons in the pigment molecule to the next energy state. because there's discrete energy states, you can't excite an electron halfway in between energy states. So you're either gonna get it to this next energy state or to two energy states. But you can't have something excited halfway in between the energy states. So that sort of explains why you can't absorb in this middle region, right? Because if you look at the wavelength of the green, it's sort of in between red and blue. And that's not enough energy to get it to a discrete level. So that gives us sort of an explanation for why you can absorb in red or blue, but not in the green. All right, so let's talk now about what is happening when you are absorbing the electrons. And I have the first picture. This is sort of a generic picture that I took from a textbook or from a website here to say generally what's happening when light strikes a molecule. So as you can see here, the photon of light hits the electron that's in the ground state, it excites it up to the excited state. So the fate is what happens when that electron goes back down. If it's at an excited state, it's not going to stay there forever because that's very unstable. So when this electron comes back down, there's three different things that can happen. The first thing that can happen, as this goes from this excited state back to ground state, is you can just give off the energy. So you're going from high energy state back to ground state. Some of the energy is going to be released. If you just let it go off, it will dissipate as heat or fluorescence, which is light. So it can be given off as heat or light. But if you do this, you're not really making use of that energy. So this is not commonly what is happening in the leaf pigments. You can, as this excited electron goes back to ground state, you can pass the energy to another pigment molecule. That's actually making use of it. And that what would happen then is that this pigment molecule would have its electron excited. And it would kind of ping pong this energy around. This is called resonance energy transfer, and this is primarily what's going to happen on this thylakoid membrane. So you're going to basically pass the electron, or not the electron, pass the energy as the electron goes back down to its ground state. The third thing that could happen is you actually fully let go of the electron. If you pass the electron to another molecule, that would be reduction, right? This would be reducing this molecule, this one would be oxidized. So you either pass the energy, which is shown in the red, or you fully pass the electron altogether. So now let's look at what is happening. Oh, sorry, that's showing fluorescence. We'll skip that part. These definitions are exactly the same as the previous slide. You can see it's given off as heat or fluorescence. This one says resonance energy transfer or reduction oxidation. Now this is putting it into the context of what is happening on the chloroplex. So just to show everybody where we're talking about, if we were to zoom in on our chloroplast right here, and we're going to show this membrane right here. This is our thylakoid membrane. On that thylakoid membrane, we have a whole bunch of chlorophyll, right? We have all these chlorophyll molecules that are embedded into the thylakoid membrane. So when light hits these, their electrons get excited, they come back down, and they pass the energy to the neighboring chlorophyll molecule. So you can kind of actually think about it like a ping pong. It ping pongs around. After the photon of light hits one, the energy is passed around the whole surface, all of these chlorophyll molecules, until it gets to a special pair of chlorophyll molecules that are called the reaction center. At the reaction center, this event happens. The electrons, instead of being just passing the energy, the whole electron gets passed. And then it's called redox. Okay, so it's going to be passed to an electron acceptor. All right. So oh yeah, and I forgot to pass this out. Let me hand these out too so we can keep track of the ICA for this one. Does anyone have questions about the fates of electrons? Or does that make sense so far? Okay. Oh, you don't, okay, okay. All right. So we're gonna come back to this, because when we talk about the light reactions, this is what we're gonna be talking about. This is how light is absorbed, and we'll talk about what happens as those electrons move. So for photosynthesis, maybe we'll just get a start on this, and then we can leave it after talking about the light reactions. Photosynthesis is happening in two stages. We said that the first stage is kind of light collection. That first set of reactions is going to do a couple of things. One, it's going to split water and produce the oxygen. The other thing that gets created during the light reactions is ATP and NADPH. These are two products that get made out of the light reactions. These two products are required for the dark reactions or the light independent reactions. So ATP and NADPH from the light reactions are going to go into the light independent reactions. And this is also called the Kalman cycle. This is what takes CO2 and ultimately makes the sugar. So that's kind of the two big steps. The first step we're going to talk about is the light dependent reactions. That is happening in our thylakoid membrane. So we're zooming in on that little square right here to talk about the light dependent reactions. So here's our thylakoid membrane. This would be the inside of the thylakoid. This is the stroma. So this is the region right outside. So here's the steps that are happening in the light dependent reactions. The first step is light is hitting what we call this antenna complex. So remember those chlorophyll molecules, just like I just showed you. They hit the chlorophyll molecules, they excite the electrons to a higher energy state, and basically they pass that energy around. So it ping pongs around in this system that's called photosystem 2. Now you might be wondering, why do we start with photosystem 2? Any guesses as to why it's called photosystem 2, even though it happens first? In science, what always happens? We name things when we discover them, right? So in the beginning, when they were first studying this, they didn't even know about photosystem 2. They only knew about photosystem 1. So this was discovered second, but it turns out that it acts first. So this thing right here is called photosystem 2. The electrons, basically the electrons hit the chlorophyll molecules. They pass that energy around until you get to the special chlorophyll pair at the reaction center. And then the electrons are passed to something called pheophytin. All right, so when they're passed, what is it called? Is it resonance energy transfer or reduction oxidation? It's going to be resonance energy transfer until you actually pass the electrons. Then it's called redux. So now the electrons are actually gone. You've passed the electrons, and that's actually the moment that this gets converted from light energy into chemical energy, as soon as you pass that electron, pheophytin. So pheophytin, you can see here, is one of those pigment molecules. It passes the electrons to plastoquinone. What does that one look like? We just saw it in our cell respiration. Quinone, does that remind you of something? Ubiquinone, right? This is very similar to ubiquinone. It's a lipid, just like it's orange here, just like the Q was in our cell respiration. It's very similar to ubiquinone. So it picks up a proton from the stroma, and it also picks up the electrons, and it is mobile. It's able to move to the second complex, which is called cytochrome complex. Now don't get that confused with cytochrome C from cell respiration. This is all very similar. But the cytochrome complex, you can see, is embedded in the membrane, and it picks up those electrons from plastoquinone. And now, because these electrons have left, we left a hole at photosystem 2. So how can we fill the hole? That's where we actually split water. So this is where water comes in, the water that is sucked up from the roots. The photosystem 2 will recover its electrons by splitting water. Okay, so we split water, that's gonna produce oxygen. Remember, oxygen is one of our products for photosynthesis. And it's also gonna give the electrons to fill the hole. So when we say for cell respiration, what was the source of the electrons? What was the thing that dropped off electrons in cell respiration? There's two different molecules. Yeah, so NADH and FADH2. In photosynthesis, the source of electrons is water. Water gives the electrons to photosystem 2. So for the blank here, we can write splitting water. There's a word for that, it's called photolysis. Photolysis means breaking by light. So photolysis is splitting water to create the oxygen, but also to fill the hole. Okay, so now where we're at so far, the electrons have been dropped off at the cytochrome complex. We can already see what's happening here is that protons are being pumped into the thylakoid space. So just like we saw for cell respiration, we're going to use a gradient. We're going to build up an electrochemical gradient, and that gradient is going to be used to make ATP. But now the purpose of ATP is different. The ATP that we're making is going to go to the carbon reactions. It's not going to be used in the cell, right? Because plant cells also have mitochondria. This ATP is going to be used for the light-independent reactions. So here on the inside, just so you kind of know where this is happening, this is where the protons are building up, inside the thylakoid space. And now we're going to let them go down their gradient. They're going to go back out through ATP synthase to create ATP. So proton flow back through ATP synthase generates ATP through chemiosmosis. And instead of being called oxidative phosphorylation, this one is called photophosphorylation, because it's light that is basically powering the buildup of the gradient phosphorylation. Okay, does that make sense? The difference between oxidative was powered by oxygen pulling those electrons through. This is powered by light. Okay. So we've gotten kind of halfway through the light reactions. I'll just finish this slide, and then tomorrow we're going to come back and review this, and we'll finish off the rest of photosynthesis. So far we've seen light hit here, bounce around through resonance energy transfer. It gets passed to pheophytin. There's the redox, and then it gets passed to plastoquinone, the cytochrome complex. Once it goes from the cytochrome complex, it needs another mobile factor, and the mobile factor is PC, which stands for plastocyanin. So PC goes in the blank. That stands for plastocyanin. Plastocyanin is kind of like cytochrome C. It's mobile. You can see that it can move, and it carries electrons from cytochrome complex to photosystem 1. Now, the electrons, once they get here, they can either be passed back into this electron transport chain by this protein called ferredoxin. So this blank should be ferredoxin. If it does that, it's called cyclic flow. You can kind of see that it makes sense. The electrons are going in a circle. They're going from PC to photosystem 1 back over here. And if you're just doing that, you're generating a lot of ATP. The other option is that ferredoxin can hand them off to NADP plus reductase, which hands the electrons to the final acceptor, which is NADP plus. So here's a very important difference between cell respiration. What was the final acceptor in cell respiration? It was oxygen. Now the final acceptor for photosynthesis is NADP plus, not oxygen. When NADP plus takes those, then it becomes NADPH. And that is the second thing that we're trying to make for the light reactions, ATP and NADPH. And this pathway, you just hand off the electrons to NADP plus, that's called linear flow because they go in a straight line. Okay, I can see that everybody's pretty toasted. So we'll come back to this tomorrow and go, make sure it makes sense, and then we'll finish off. We only have about 7 more slides for photosynthesis. Thank you for bearing with me.